In this second video of the respiratory system, we're going to discuss the other three ways in which the respiratory system carries out gas exchange. So we did a pretty thorough treatment of ventilation with the first video, which is moving air into and out of the lungs. And so now we're going to talk about pulmonary gas exchange. We're going to talk about gas transport in the bloodstream, and we're going to talk about peripheral gas exchange. Okay, so I'm going to do something terrible to you right now and introduce physics. And there are a couple of different gas laws that you're going to have to know. The first is Dalton's law, which says that in a mixture of non-reacting gases, the total pressure exerted is equal to the sum of the partial pressures of the individual gases. Now that is quite a mouthful to unpack. So let's take the first part of the sentence, in a mixture of non-reacting gases. So this would be a mixture of gases that don't chemically react with one another. And so the atmosphere would be a really good example of that. You've got nitrogen and oxygen and argon and CO2, and they don't chemically react with each other, so that means that they are non-reacting gases. So what we're going to do is we're going to go through Dalton's law, and we're going to use the atmosphere as kind of our model for this. And so the atmosphere, the total pressure of the atmosphere is 760 millibars. That's something that we saw in the first video. Now, when you're talking about partial pressures in this gas mixture, then the percentage of the gas, the concentration of the gas, is also going to be the percentage of its partial pressure. So by way of explanation, let's just kind of work our way through this. So the atmosphere is 78% nitrogen. Okay, and our total pressure for the atmosphere is 760. So what is our partial pressure of nitrogen in the atmosphere going to be? It's going to be 78% of 760. So our partial pressure of nitrogen, because the atmosphere is 78% nitrogen, our partial pressure of nitrogen is 592.8. Our atmosphere is 21% oxygen. And so what is 21% of 760? That's going to be 159.6 millibars. So 159.6, if we round it up, it would be 160. That is our partial pressure of oxygen in the atmosphere. Now, if we take a look at our other major respiratory gas, which we're going to have to, it's CO2. Now, CO2 is 0.04% of the atmosphere. And so what is 0.04% of 760? It's about 0.3 millibars. And so if we take in just the atmosphere, if we just take in air, if we're breathing in air at normal air pressure at sea level, we would expect to be taking in about 160 millibars partial pressure oxygen, about 0.3 millibars partial pressure CO2. Now, if the percentage of these gases changes, then their partial pressure changes. Now, the pressure of a specific gas in a mixture is its partial pressure. So, in the atmosphere, 159.6 or 160 would be the partial pressure of oxygen in the atmosphere. Okay? Each gas in a mixture of gases exerts its own pressure as if no other gases were present. Okay, so if you have 159.6 or 160 millibars of oxygen partial pressure, if you have a different gas mixture that is also 760, but let's say that the partial pressure of oxygen in gas mixture number 2 is 50 millibars, then you're going to see oxygen flow from our atmosphere to that other gas mixture. Okay, so PO2 means partial pressure of oxygen. PCO2 means partial pressure of carbon dioxide. Now I'm going to come back to that example about the movement of oxygen. Okay, so gases will diffuse from an area of higher partial pressure to an area of lower partial pressure. Now, I did say in the last slide that the higher the percentage of gas in a mixture, the higher the partial pressure. So you can kind of think of partial pressure as the equivalent to concentration. So things diffuse from an area of higher to lower concentration. With gases, the way that you measure concentration is with partial pressure. So gases will go from higher partial pressure to lower partial pressure. Now, one thing that I said on the last slide that I hope to kind of clear up now is each gas in a mixture exerts its own pressure as if no other gases were present. So let's take a look at this. Now, if this is two gas mixtures on the left and on the right, in this scenario, the gas mixture on the left and on the right have the same total pressure. They have the same total pressure. Now let's see with this gas mixture though. You've got three gases in here. One is depicted by blue, one by red, and one by gray. And so just looking at this, let's take the blue and the gray dots out and just see what the red dots look like. And so you can see here that there's a higher amount of gas, red gas, on the left than on the right. So there's a higher partial pressure of red gas on the left than on the right. So that means that even though the total mix has the same total pressure, still the red gas is going to diffuse from left to right because it acts as if no other gases were present when with respect to partial pressure. Let's take a look at the gray gas. So you can see that there's a higher concentration of gray gas on the right than on the left. Therefore, a higher partial pressure of the gray gas on the right than on the left. So red gas is going to move to the right, gray gas is going to move to the left, blue gas may remain there, may remain the same. But because the red gas is at a higher concentration on the left than on the right, it's going to move from left to right. So it's going to move from its area of higher partial pressure to its area of lower partial pressure. Now, just as with concentrations, if there's a greater difference in partial pressure, there's a faster diffusion. Just the same way that if there's a greater difference in concentration in liquids, diffusion goes faster. So the greater the difference in partial pressure, the faster the rate of diffusion. So which of these, the red gas or the gray gas, do you think would diffuse faster? My guess would be the red because I think there's a little bit more red on the left, there's gray on the right. So when it comes to pressures, let's make this really clear, okay? Total pressure makes air move in and out of the lungs. So ventilation is about increasing total pressure, up or down, okay? So moving from 760 to 759 or 761, okay? So total pressure makes air move in and out of the lungs. The entire gas mixture moves, okay? So what that means is that since the entire gas mixture moves, the proportions don't change. Now, partial pressure determines how fast the individual gases diffuse across the membranes once you're in the respiratory area of the lungs. So total pressure moves the gas mixture to the respiratory areas. Once you're in the respiratory areas, then you have to think about partial pressure, which will help the gases diffuse across the cell membranes. Okay, so we will look at this in more detail. Okay, now this law, the difference in partial pressures, is not the only factor that affects diffuses of gases into the bloodstream. So I want you to remember, it's air in your lungs, it's liquid in your bloodstream. So gases are moving from air to liquid. And so this brings up another law called Henry's law, and that is the amount of dissolved gas in a liquid is proportional to its partial pressure in the gas phase. So if you have a higher amount of red gas in the air mixture, if you look at the blue portion, which is supposed to be water, you're going to have a higher amount of red gas in the liquid. Less gas in the air, less gray gas in the air, less gray gas in the liquid. So that's proportional. Now, solubility into water also plays a role. So I want you to think about this. Different solids dissolve into water at different levels. So think about protein powder, very difficult to get to mix into water, right? Compared to that, salt, table salt, really easy to get to dissolve into water. So solids will dissolve into water at different rates. Gases are not any different. Different gases will dissolve into water at different rates. So they have different. So with the solubility into water playing a role, let's take a look at oxygen and CO2. As it turns out, CO2 dissolves into water much more easily than oxygen, and in fact, the solubility of carbon dioxide is 24 times the solubility of oxygen. So you can dissolve 24 times as much CO2 into water as you can oxygen. So this is the reason why we've got carbonated drinks, but we don't have oxygenated drinks because it's a whole lot easier to put CO2 into water than it is oxygen. Now let's take a look at gas diffusion. Okay, so again, CO2 dissolves into water much more easily than water. And okay, so let's take a look at oxygen diffusing around the system. So when it comes to diffusion of oxygen in the lungs, the partial pressure in the alveoli is going to be, you can see it on the top right, 104 millibars. But we have to start out by looking at the inspired air on the top left. So you can see that the highlighter there has got our partial pressure of oxygen at 160, and that's what we calculated it as in a prior slide. Now, when the air that you breathe in moves into the lungs, there's going to be air that's already there, and that air is going to have a good bit of CO2 in it. And so that mix means that your partial pressure of oxygen, because now it's less of the percentage of air, is going to be at 104. So the partial pressure in the alveoli is going to be 104 millibars. Now, this will diffuse into the blood that's coming into the capillaries. So the blood that leaves the lungs in the capillaries in the alveoli is 100 millibars. You can see that highlighted in the area below. Now, when you get to the systemic tissues, the partial pressure in the capillaries out there is actually still 100 millibars. You haven't had any oxygen loss out of these arteries on the way down or on the way there. Now, those tissues that you're taking the oxygen to, they've been using oxygen. And so they're going to have less oxygen and more CO2 because they've been consuming oxygen and generating CO2 as a part of cellular respiration. This means that the partial pressure of oxygen in the tissue cells is actually less than 40 millibars. Now, this is actually a good thing for us because there's got to be a big difference in partial pressure to move the oxygen from the blood into the tissues. And so the fact that oxygen doesn't dissolve well into liquid means that it's difficult to move it around in liquid. So in order to get oxygen to move, you've got to have this great big difference between partial pressures. Now, if we go on the way back up, the blood that leaves the tissues and the lungs, the blood that leaves the tissues and lungs, look at the bottom left, the PO2 is at about 40 millibars. And so basically you've had a lot of oxygen unloaded from that blood into the tissues. Now, when this blood gets back up to the alveoli, so when that blood makes it back up to, I'm going to use blue for deoxygenated blood, when that blood makes it back up here to these capillaries, it's still going to be at a PO2 of 40 millibars. So what's the PO2 of the air in the alveoli? It's 104. So that means that oxygen will diffuse from 104 to 40 and give you a final sort of number of 100. So again, what are the differences? At the alveoli, the gas exchange, you have to have this difference of 64 millibars to get oxygen to move. At the tissues, you have a difference of at least 60 millibars. And so with that big a difference, even a reluctant mover like oxygen can be moved. Now let's talk about CO2. Now I want you to remember that CO2 can dissolve into water 24 times more easily than oxygen, so it's going to be a lot easier to make it move. So let's see because you generate CO2 in the tissues, let's start at the bottom right with the tissues, CO2 in the systemic tissues. So your partial pressure of CO2 in the systemic tissues is going to be, in the systemic capillaries is going to be less than 40 millibars. So there's not a lot of CO2 in the tissue capillaries at all. Now, in the tissue cells themselves, despite the fact that they've been generating CO2, they still haven't raised that PCO2 number up much. So the partial pressure of CO2 in the tissue cells is going to be a little bit greater than 45 millibars. And so there's just a 5 millibar difference. But because CO2 moves through liquids so much more easily, this small difference of 5 millibars is enough to make the CO2 move from the tissues into the bloodstream. And so what that means is that in the alveoli, the partial pressure of CO2 is going to be 40 millibars. In the capillaries that lead into it, the partial pressure is going to be 45. So when this deoxygenated blood that's entering here comes into the lungs, it has this PCO2 of 45 millibars. The PCO2 of the air that's present in the lungs is going to be about 40. So there's just a 5 millibar difference in pressure, yet the CO2 will move from that deoxygenated blood into the airspace of the lungs. So remember that oxygen required this giant difference in partial pressure, around 60 millibars, and CO2 only requires a small one. So the reason why CO2 only requires this small difference in pressure to move is because its solubility in water is a lot better than oxygen. So again, much easier to move CO2 back and forth. So if we take a look at what happens in the alveolus, you've got a number of different factors that affect the gas exchange. So the partial pressure difference between the gases is a factor. The surface area that's available for gas exchange is also a factor, and our lungs have got a tremendous amount of surface area available for gas exchange. So if you were to take out a human lung and spread it out on a tennis court, the type 1 pneumocytes, the respiratory exchange cells, would cover half of a regulation tennis court. So there's a tremendous amount of surface area available for gas exchange. Now, if you reduce that surface area because of damage to the lungs, then you can run into trouble with gas exchange. Okay, so again, any disease that decreases surface area slows the rate of gas exchange. So tuberculosis, there's a lot of scar tissue that's formed in TB. Anything that causes fibrosis or scar tissue is going to slow the rate of gas exchange. So emphysema is a good example of this as part of COPD. Diffusion distance is another factor in terms of gas exchange. So if this capillary and alveolar membrane becomes thicker, and let's try a different color here. So if the membrane here becomes thicker, if there's scar tissue instead of these cells, it becomes thicker. Anything that increases this diffusion distance, like an increased amount of fluid in the lungs, you're going to overall increase your diffusion distance here. You increase diffusion distance, you make diffusion harder. And again, because it's so difficult to get oxygen to dissolve into the bloodstream, if you increase diffusion distance, you're going to impair your oxygen intake pretty dramatically. Now, the molecular weight and the solubility of the gases, this plays a role as well. Now, oxygen does weigh a little bit less than CO2, so if they had the same solubility, then oxygen would probably diffuse faster because it's a little bit smaller. But the solubility kind of overrides that, and it means that oxygen moves much more slowly into liquid and through liquid than CO2 does. Okay, now, because oxygen dissolves really poorly into liquid, you can't just dissolve it straight into the bloodstream for transport, because a sufficient amount for your tissue needs simply could not be carried. So one thing that I need to tell you about gases and dissolving in the bloodstream is this. So it's just the opposite of solids when it comes to temperature. So when you make sweetened tea, you want to stir the sugar in when the tea is really hot because it's easier to dissolve sugar solids into liquids at higher temperatures. With gases, it is just the opposite. A higher temperature means that gases cannot dissolve into the bloodstream as fast. Even CO2 is going to have its rate of dissolving into the bloodstream decreased. I want you to think about what happens versus a warm Coke versus a cold Coke. So if you shake a warm Coke, it'll fizz up a whole lot more than if you shake a cold Coke. And that's because in a warm Coke, the CO2 that's in the Coca-Cola comes out of solution faster because warm liquids don't dissolve gases as easily. So the high temperature of the bloodstream affects the transport of oxygen. So it's a poor dissolver into liquid in the first place, but given the fact that your bloodstream is at about 100.3 degrees Fahrenheit, oxygen is really not going to want to dissolve into there. So basically you've got to bind oxygen to hemoglobin in order to transport it. So you've got to do this to carry enough oxygen for your tissue needs, and you've also got to do this to keep it from coming out of solution. I mean, having oxygen bubbles floating around your bloodstream is going to be a really bad idea. Okay, 98.5% of oxygen gets transported attached to the hemoglobin molecule. So there's hardly any dissolved oxygen circulating in your bloodstream. Now, the higher the partial pressure of oxygen, the more it will bind with the heme portion of the hemoglobin molecule. And so what's the heme portion? It's actually going to be these green areas right here. So you see 4 of them on this molecule there. There's another one here. There's another one here. And number 4 is really kind of hiding back behind here. So 4 heme molecules, each one can carry an oxygen molecule. Okay, so if a hemoglobin is carrying its maximum, then it's saturated. Okay, and this leads us to another measure called the percent saturation of hemoglobin. This is your O2 saturation. If you've ever had like one of those little clips that they put on your finger to measure your O2 saturation, this is what they're measuring. What percentage of your hemoglobin is full up on oxygen? And so the O2 saturation is the average saturation of hemoglobin with oxygen. Now there are some things that can change the amount of saturation in hemoglobin. When the PO2 is high, hemoglobin binds with a lot of oxygen, it gets almost 100% saturated. When PO2 is low, because there's not a lot of partial pressure diffusing oxygen into the bloodstream, then hemoglobin will only get partially saturated. But fortunately, hemoglobin is so great at picking up oxygen that even with a PO2 of 40 millibars, hemoglobin saturation remains as high as 75%. So basically, hemoglobin is really good at sticking to oxygen, so that even with oxygen refusing to diffuse well into water, even at a lower PO2 of 40, that's 1/4 normal atmospheric pressure, that's 1/4 of normal atmospheric oxygen partial pressure. Even at 25% of normal partial pressure, you can still saturate 75% of your hemoglobin. Okay, so now we're going to look at a graph of hemoglobin saturation in different locations of the body, and they can be different because different locations of the body can have different oxygen needs. So if a tissue consumes more ATP than it needs more oxygen, if it consumes more oxygen, then the saturation is in those tissues is going to be lower. So let's take a look at our axes here. So this axis actually is the partial pressure of oxygen. And so that partial pressure is going to go from 0 to a little bit over 100. This axis is the percent saturation of hemoglobin, and of course that can only go up to 100. Okay, now with that in mind, let's take a look at some of the data on this graph. Now, this is deoxygenated blood that is in contracting skeletal muscle. So skeletal muscle uses a whole lot of oxygen, and it means that the PO2 in the blood that's in skeletal muscle is going to be very low. Your partial pressure is going to be about 20 millibars. And at 20 millibars, the percent saturation of hemoglobin is at about 35%. You see where the dotted line is. Now, your average deoxygenated blood in systemic veins, this is going to be blood that's coming out of high demand tissues like skeletal muscle and low demand tissues like adipose. And so it's going to average out to a PO2, a partial pressure of about 40 millibars. Now if you follow that line, that purple dotted line up, you can see that you've got about 75% saturation of a hemoglobin. So just to kind of summarize here, at this very low partial pressure of 20, about 35% of your hemoglobin is full up. At this lower pressure, or at a higher pressure, but still really low, of 40 millibars, then you've got about 75% of your hemoglobin that's full up. Now let's consider oxygenated blood in systemic arteries. So this is the blood that's coming out of your lungs. So your PO2 is actually going to be a little bit beyond 104. And this means that your percent saturation is going to approach 100. It's not quite at 100, but it's pretty close to it. So the more ATP, again, that's used in a tissue, the more that it soaks up oxygen, and the more that oxygen it pulls out of the blood that's around it, which means that that blood has a lower partial pressure of oxygen. So high demand tissues have low PO2s in the blood around them. Lower demand tissues they've still got fairly low PO2s because they use oxygen too, but it's not as low as the high demand tissues. And finally, the blood that's coming out of the lungs is going to be a high PO2, therefore a really high saturation. So again, the more ATP that a tissue consumes, the higher the need for oxygen, the lower the saturation of oxygen in the tissues. Now, let's examine the implications of this, okay? So, in exercise, a resting tissue, let's take a look at resting tissues first. So, resting tissues have a PO2 of about 40 millibars. Now, this means that they're going to have a saturation of about 75%. You can see this from this graph. Now, if we look at the amount of O2 that's present, or the saturation that's present in arterial blood, it's up here at close to 100. And so it means that there's this much difference between the saturation of the arterial blood and the saturation of these resting tissues. So that means that you can unload oxygen to resting tissues because of the lower PO2 and the lower saturation in the resting tissues. Now let's take a look at exercising tissues. And so exercising tissues can have a PO2 as low as 20. And so let's see how that corresponds with our graph. So again, about 25% saturation. So look at the y-axis, you can see it's 25% saturation. Now, considering the O2 saturation in arterial blood, you've got this much of a difference between the arterial blood and the exercising tissues. So that big a difference means it's a lot easier to unload oxygen onto exercising tissues. So one of the cool things about this is the lower the oxygen that you have, the more that you need it, the easier it is for you to get it. So um, high demand means quick unloading. Now, like any protein, hemoglobin folds, and the way that it folds can be affected by physical conditions. And so what are the physical conditions that can affect how hemoglobin folds? Differences in pH, differences in CO2 concentration, this can affect how hemoglobin folds. Now, there are two different arrangements of hemoglobin. In one of them, hemoglobin, in the relaxed form, which you see to the right, hemoglobin has a higher affinity for oxygen. It means that it sticks to oxygen a lot better. You could think of this hemoglobin as superglue for oxygen. So in this relaxed form, it will pick up oxygen very easily. So it's easy to pick up oxygen in the lungs. But if you get this relaxed form out to the tissues, it's a lot harder to unload them to the tissues. Fortunately for us, there's another arrangement for hemoglobin, and this is called taut. And so this, this arrangement of hemoglobin has a lower affinity for oxygen. That means if you get this taut form of hemoglobin close to the lungs, it's harder to pick oxygen up, but it is easier to unload oxygen to the tissues. So this is the taut form of hemoglobin. Now let's examine how the differences in both pH and temperature can make a difference in how the hemoglobin. So let's take a look at this diagram, and the first thing that I want to do is sort of reorient you to what we're looking at. So the x-axis is going to be your PO2. Your y-axis is going to be the percent saturation of hemoglobin. Now let's take a look at normal circumstances. So at a normal arterial CO2, then you have a PCO2 of 40, and you have a pH of about 7.4. Now, if you increase the CO2, you're going to decrease the pH, and that's going to change how hemoglobin folds. And so let's go ahead and make those changes. So we increase the CO2, we decrease the pH, and our hemoglobin starts to change. And so if you'll recall, I said that this taut form has less of an affinity for hemoglobin. It means it does not pick up, it has less of an affinity for oxygen. It does not pick up oxygen as easily. And let's examine the difference here. So if you're talking about a decreased pH, if you're at a PO2 of 20, your percent saturation is going to be about 20% saturation. So because hemoglobin in its taut form does not hold on well to oxygen, it won't pick up as much. So at a PO2 of 20, it has a percent saturation of about 20. Now let's take a look at the normal mix of hemoglobin. So at a normal mix of hemoglobin, there's more relaxed form than there is at the lower pH. And so what this means is that overall, your bloodstream, the hemoglobin in your bloodstream, is on average going to have a higher affinity for oxygen. And it means that at a PO2 of 20, you're going to have more saturation of the hemoglobin. So let's step back for a second and realize what this says again. So at the same pressure, partial pressure of oxygen, at the same diffusion rate, taut hemoglobin picks up less oxygen than regular hemoglobin. Okay, so the lower pH mix is going to have more taut hemoglobin. The normal arterial mix is going to have a standard amount of taut versus relaxed hemoglobin. So more relaxed means more affinity, which means a higher saturation at the same partial pressure of oxygen. Now let's take a look at what happens. Um, this is basically the same thing, that an increased carbon dioxide results in increased hydrogen ion concentration. And this diagram is the reason why. So, or the equation is the reason why. So when you combine CO2 and H2O, it's going to make carbonic acid, and that is going to decrease the pH. That's the reason why increased CO2 means a decreased pH. That decreased pH will cause hemoglobin to fold differently. Now let's say that you have a decreased CO2. If you have a decreased CO2, look at, this is the normal line. Now let's decrease the CO2. If you look at the blue line, the CO2 is lower, which means the pH is higher, which means that more hemoglobin is in the relaxed form. So you can see that this curve has shifted to the left. What this means is that now, under these conditions, blood hemoglobin has a higher affinity for oxygen. So let's take a look at our values again. So we're going to go at 20 again. So with a normal amount of CO2 at a partial pressure of 20, you get 40% saturation. If you have low CO2 and alkaline pH, then at 20 partial pressure, you're going to get over 60% saturation. This means that hemoglobin is sticking to oxygen very well. Now, where this becomes relevant is that in the area of the systemic tissues, there's a lower pH because tissues consume oxygen and produce CO2. That makes the CO2 at the tissues lower. That means that you're in the taut form of hemoglobin. At the tissues, you can unload oxygen much more easily. Now, as you move back to the lungs, the area of the lungs has much less CO2. There's going to be a higher pH. Your hemoglobin will shift more to the relaxed form. You can pick up oxygen much more easily. Now, this is something in biology that I think is just about miraculous. So, in the lungs, you want to pick up more oxygen at the lungs. That's the whole point of being at the lungs in the first place. At the lungs, higher pH, relaxed form of hemoglobin, greater pickup of oxygen right where you need to pick up more oxygen. When you move out to the tissues, your pH drops because of all the CO2 out there. When your pH drops, you shift to the taut form. Your hemoglobin does not hang on to oxygen nearly as easily, and this makes it easier to move the oxygen from your red blood cells to the tissues that need it. So the reason why hemoglobin is found in a lot of different organisms is because it is spectacularly good at what it's supposed to do. In an oxygen-rich environment, it picks up oxygen. In an oxygen-poor environment, it lets it go. So basically, hemoglobin changes forms between the lungs and the tissues in a way that really suits the needs of gas exchange. Okay, now temperature will do something fairly similar. So at a higher temperature, you'll see hemoglobin in the taut form. So this curve shifts to the right. So at a normal body temperature of 38 C, you're going to see a certain affinity of blood hemoglobin for oxygen. Okay, so at a PO2 of 20, you're going to have about 25 or 30% saturation of hemoglobin. Now, if we raise the temperature, at the same partial pressure, you've got less than 20% saturation. So basically, higher body temperature means that your more hemoglobin shifts to the taut form, and it means that hemoglobin has less of an ability to pick up oxygen. So this is one of the reasons why when your body temperature rises with either fever or exercise, your respiration rate also has to rise in response. You've got to breathe faster to try to drive more oxygen into your bloodstream because at a higher body temperature, your hemoglobin has less affinity for oxygen. Now, there is one thing I do want to say about this diagram, and it's that the temperatures are really kind of exaggerated for effect to show the difference between the curve more, because 43 degrees Celsius is about 109 degrees temperature, and if your temperature is at 109, you got bigger things to worry about than your hemoglobin saturation. Now, let's just do this for 80 to get kind of a contrasting viewpoint. So 80 is really more of a normal type of PO2 concentration or partial pressure. And so at a normal body temperature at 80 millibars, you're going to have about 90% saturation. If you've got this massive fever of 109 degrees, at 80 millibars, you're only going to have like 81% saturation. But like I said, your bigger problem is the fever, not so much your hemoglobin saturation. So a decreased temperature will result in hemoglobin adopting the relaxed form. And unfortunately, this is where I have to tell you about an error in the PowerPoint that's online in eLearn. That says at a higher temperature. At this next bullet point, that should say at a lower temperature. So at a lower temperature, more hemoglobin is in the relaxed form, meaning that, again, this hemoglobin has a higher affinity for oxygen. It sticks to oxygen better. So this is like your oxygen superglue. At the high temperatures, at the low pH, it's like a post-it note, oxygen post-it note, not a very strong lube. So let's take a look at different PO2s. So at a PO2 of 80, at normal body temperature, you're going to have about 90% saturation. If your body temperature has dropped to 68, again, you've got much more problems than your hemoglobin affinity, but let's run those numbers anyway. You're going to have about a 95% saturation level. Now, at 10 degrees Celsius, which is about 50 degrees Celsius again, you're in a lot more trouble than your hemoglobin saturation. 100%. And in fact, at 50 degrees, even at a 42, a PO2 of about 42, you're going to get 100% saturation. Now, you may have heard stories of people who have drowned at really, really low temperatures. And when their body temperature drops, it does mean that what little oxygen they were able to take in is really going to stick to the hemoglobin at a lower body temperature. So the other thing that happens in these is that you don't get the generation of a lot of free radicals. Everything slows down. But this hemoglobin affinity, this can be one of the reasons why you can see some survival of drowning victims under very low temperatures for longer times. Okay, so now that we've talked about oxygen transport and beaten that to death, let's talk about CO2 transport. And this is something that's going to happen completely differently from hemoglobin. Okay, so the first thing to tell you is that 9% of it is just dissolved in the blood plasma. So even though CO2 has this really great rate of dissolving, 24 times that of oxygen, blood is so hot that it's really impractical to carry CO2 as a dissolved gas in blood plasma. 13% gets bound to hemoglobin as carbaminohemoglobin. Okay, this is, well, I'll tell you why it's termed carbaminohemoglobin in just a little bit. 78% of it gets transported as bicarbonate ions. So that chemical formula that I showed you earlier today, or earlier in this video, it comes into play here. So basically, CO2 and H2O will form carbonic acid, and carbonic acid will separate into the bicarbonate ion and the hydrogen ion. So The HCO3, the bicarbonate, can bind to plasma proteins. But this kind of transition from CO2 and H2O into carbonic acid doesn't happen quite fast enough for the body's purposes. And so what you'll see is an enzyme called carbonic anhydrase that will increase the rate of carbonic acid production. And again, carbonic acid is H2CO3. So carbonic anhydrase will increase how fast that CO2 and H2O combine to make H2CO3. Okay, so let's talk about carbaminohemoglobin. So the first thing to tell you is that this sort of light blue, these light blue ribbons, these represent hemoglobin amino acid chains. So this doesn't represent the heme portion of hemoglobin. So let's stick that in there. These are the heme groups, and these are the groups where oxygen is bound. So the red area binds to oxygen. Each one of those red areas can bind to one oxygen molecule. Now, where is it that CO2 binds if hemoglobin is carrying it? It doesn't actually bind to the heme groups. Instead, it binds to some amino acids. So there are 4 amino acids around the hemoglobin molecule, which are pictured in black, and these will bind carbon dioxide. And because the CO2 is bound to amino acids, this is the reason why it gets called carbaminohemoglobin. So let's take a look at carbonic anhydrase a little bit more closely. So this is the reaction that is catalyzed by carbonic anhydrase. Basically, CO2 and H2O is combined into carbonic acid. Now, I want to make something clear, and it's that carbonic anhydrase is contained within red blood cells. So the CO2 has to enter the red blood cell before carbonic anhydrase will act on it. But it does diffuse into red blood cells very easily. So once the CO2 is in the red blood cell, carbonic anhydrase will combine it with water that's there and form carbonic acid. So how does this bicarbonate ion actually make it out into the blood plasma? So if you'll recall, I did tell you in an earlier slide that 78% of CO2 was carried in the blood plasma as the bicarbonate ion. And so what is it that happens? So you have an antiport protein that will exchange carbonic acid for chlorine. This is called the chloride shift. So let's watch it in action. So the chloride ion gets brought into the red blood cell. The bicarbonate ion gets moved out of the red blood cell, and now you've got the bicarbonate ion in the bloodstream. Okay, now what happens to the hydrogen that's generated? So the hydrogen doesn't just sit there in the red blood cells. So at this point, you've got hemoglobin, which is in the red square at the bottom that are just highlighted, hemoglobin that is deoxygenated, and the hydrogen, which is in that sort of purplish square up above it. So these two are going to combine, and hydrogen is going to reduce the deoxygenated hemoglobin. And so you have this reduced hemoglobin. So this is how the hydrogen ion is actually kind of carried in the red blood cells here without dropping the pH within the red blood cells themselves. Okay, now the lower that the PO2 is and the lower that the hemoglobin saturation is, the more CO2 can be carried in the blood. And the reason why is because reduced hemoglobin is better able to bind to CO2 and form carbaminohemoglobin. So that HHB, that sort of final product that you get from the combination of hydrogen and hemoglobin, can better bind CO2 and can better form carbaminohemoglobin. Now, if you've got tissues that have more CO2 produced, then this will tend to cause oxygen to dissociate from hemoglobin. So you see the big red arrow moving down. So at this point, oxygen dissociates from hemoglobin, it gets unloaded, and will move into the systemic tissues. On the other hand, when you're at the lungs and you have a high oxygen content, CO2 gets bumped off of hemoglobin and gets exhaled. But there's more that happens here. So you'll recall that carbonic anhydrase actually joined CO2 and H2O to make carbonic acid. Now the remarkable thing is at the different pH, at the higher pH that you find around the lungs, the carbonic anhydrase actually reverses its action. So it will take carbonic acid and split it into CO2 and H2O. At this point, the CO2 diffuses out of the red blood cell into the alveolar space because the alveolar air has a lower partial pressure of CO2 than you find in the bloodstream. So the carbonic acid will reverse its action. Now, in order to get H2CO3 to transform to CO2 and H2O, you've got to have the bicarbonate and the hydrogen come from somewhere. So you're actually going to see a reversal of this antiporter. You're going to have a chloride shift out via a transport protein, and so chloride goes out, bicarbonate comes in. So this is where you get the bicarbonate to form the carbonic acid. And where is it that you get the hydrogen from that you need to combine into carbonic acid? Well, the reduced hemoglobin, the oxygen that's there will actually knock the hydrogen off of the hemoglobin, and so you'll have oxygenated hemoglobin and hydrogen. And so this means that the hydrogen now is available to be combined with the bicarbonate to form carbonic acid. And so you really do see a reversal of what happens in the lungs versus what happens in the systemic tissues. Now, I talked about how remarkable that hemoglobin was for changing its form between the lungs and the systemic tissues. Easy to pick up oxygen in the lungs, easy to release it at the systemic tissues. Carbonic anhydrase does a similarly remarkable shift in function. So at the tissues, it picks up CO2 and H2O, combines it into carbonic acid so that you can get that bicarbonate ion put out into the bloodstream for transport. Now the nice thing about transporting it that way is that bicarbonate is a solid. It's not a gas. And so there's no chance of the bicarbonate bubbling out of the bloodstream like CO2 possibly could. Think of what happens again to a warm Coke when you shake it. It fizzes right up. All those bubbles of CO2 coming out of solution. Now imagine that if you were carrying a bunch of CO2 gas in your bloodstream, you shiver and or you shake your arms around, you might fizz up your blood, and that's going to be a really bad look because those gas bubbles are going to start blocking capillaries. So this is a way of transporting CO2 without transporting it as a gas. So the reversal of carbonic anhydrase is really important. Again, it combines the CO2 into carbonic acid, which you can then split out as bicarbonate and hydrogen. But at the lungs, it's going to take the available carbonic acid, split it back out into CO2 and H2O so you can get rid of the CO2. So the fact that it reverses its actions because of a different pH, I think is something that's really remarkable. Okay, so let's talk about how all of this stuff gets controlled. The respiratory centers of the brain. I probably should have covered this with ventilation, but let's talk about this anyway. And so the pons is going to contain what's called the apneustic center. And what this will do is it'll stimulate inspiration by stimulation of the dorsal respiratory group, which is going to be right here. So here's the dorsal respiratory group. Okay. Now, the pneumotaxic center of the pons will inhibit the activity of both the dorsal respiratory group and of the apneustic center. And so you're not stimulating inspiration, you're inhibiting inspiration. And so this pneumotaxic center will also initiate the process of forceful exhalation. And so pneumotaxic kind of translates into air moving. And when does air move faster than when you're forcefully exhaling? So that name should make a little bit of sense. There's a respiratory center in the medulla, which is called the there's a ventral respiratory group and a dorsal respiratory group, both in the medulla, that establish the rate and the depth of breathing. So the medulla and the pons kind of combine to determine how fast and how deep you're breathing, and whether you're stopping or starting breathing above and beyond the normal level of tidal volume. So let's talk about things that can increase or decrease the rate of breathing. So first thing, this is your respiratory center in the medulla and the pons, and let's examine what it is that can increase and decrease your rate of breathing. So higher brain centers, the cerebral cortex, you can exert voluntary control over your breathing. You can breathe faster or slower at will, and so this is one of the things that can exert control over breathing rate and depth. Other receptors like pain and emotional stimuli. If you get really upset or angry, you start breathing faster. This acts through the hypothalamus, and pain can make you breathe faster as well. So other receptors and emotional stimuli can cause you to breathe faster or slower in an involuntary fashion. You've also got peripheral chemoreceptors, and these peripheral chemoreceptors, if you have a decrease in oxygen or an increase in CO2, or an increase in hydrogen, and this will cause an increase in breathing rate, because basically a higher amount of hydrogen can be an indicator of a higher amount of CO2. And so you really want to try to get rid of CO2 as much as possible. It's really toxic for you, so you're going to want to increase your rate of breathing to increase your rate of expiration. Now, decreased oxygen, if you're low on oxygen, you got to take more in. So these chemoreceptors can increase your breathing rate by detecting low O2, high CO2, and high pH. Now, you've got central chemoreceptors, which, curiously enough, don't respond to a lack of oxygen, but they will respond to an increase in hydrogen or a buildup of CO2 by making you breathe faster. Now, there are receptors in muscles and in the joints. These can increase your rate of breathing. And basically what's happening here is, if your body position is changing rapidly, then that information gets fed back into your central nervous system. And so if you're changing position rapidly, more than likely you need more oxygen, so you want to increase your breathing rate. If you have the lungs being stretched out a whole lot, the stretch receptors in the lungs can actually decrease your rate and depth of breathing. So basically, this is when your lungs say, okay, we've breathed deeply and hard for long enough, and we need to slow down our breathing rate and our depth of breathing. Irritant receptors can also decrease the rate of breathing. So I mentioned that irritants can cause bronchoconstriction in the previous video. But these irritant receptors will also lower your rate of breathing. So basically, you're trying to protect your lung tissue from irritants, so you're going to do two things. You're going to slow down how much you breathe, so you take fewer breaths, bringing fewer irritants in. And you're going to squinch down your bronchioles so that you can't quite get as much of that irritant-laden air into your respiratory surfaces per breath. One final thing, when CO2 levels in the bloodstream rise, this is called hypercapnia. It's going to trigger a breath. So when you hold your breath, that overwhelming urge to breathe, that urgency to breathe, is caused by increased CO2 levels. Because although decreased oxygen levels can trigger breathing, it's not nearly as big a stimulant for faster breathing than CO2 level. So I'm going to tell you one more thing. Have you ever been at a party and breathed helium out of helium balloons so that your voice sounded squeaky, and then you get a headache? Now that headache is oxygen deprivation, comes from oxygen deprivation. And so you're thinking, if you're low on oxygen, why didn't you feel an urgent need to breathe? The reason why is because, again, decreased oxygen levels don't stimulate that urge to breathe nearly as much as high CO2 levels. So you're breathing that helium, you're still getting rid of CO2, so you don't have the urgency to breathe. So that's the reason why you continue to inhale the hydrogen I'm sorry, the helium. Crap. You continue to inhale the helium. You're still getting rid of CO2, but you're not getting enough oxygen in. And so you don't feel the urgency to breathe, but yet you get low on oxygen levels. Bang, headache.